All right, so we're going to start off the week by starting our notes on cell cycle and mitosis. If you haven't already taken your test, make sure you go and take your test right now, and then come back to these notes anytime today, just because obviously the test is kind of important and it is the first big grade of your new marking period. So starting off, I would love it if you could write this down, even though it does say do now. So brainstorm what you remember, if you remember anything from biology. So there are three main reasons why cells replicate and divide. See if you can list them for me. Take a pause and write those down. And our actual main reasons why they replicate and divide in the first place is because organisms that have multiple cells need to repair the body when cells get damaged. Cells replicate and divide also during reproduction, but that one's actually a separate one that we'll talk about at a different point in a different chapter. And then we have replace and growth. And so when cells die or when cells cease to be able to function, they need to be replaced. So therefore, cells need to duplicate themselves by splitting in half in order to replace the ones that are no longer functioning. And then growth, increasing the number of cells is how organisms grow larger, right? The cells don't actually get that much bigger. We talked about the issue with that when we talked about surface area to volume ratio, right? When the surface area to volume ratio isn't high enough, cells actually can't transport the materials in and out of the cells. For example, like wastes, like carbon dioxide, right? So that means that cells, instead of just growing larger continuously, at some point they'll stop growing larger and they'll actually split off into two smaller cells, and those cells will grow a little bit before they split into 4 smaller cells. So growth is actually based on how many cells you have and not actually how large your cells are. So the second thing I want you to think about, so I want you to write the question down and then I want you to pause and try to think of the answer. So what are 2 things? You can just list one if you can't think of 2, but what must cells do before they divide? So the answers are that DNA must be replicated, right? You're not going to split the cell and have 50% of the DNA in one, 50% in the other in regular mitosis cell cycle, right? Again, it's a little bit different when you talk about meiosis and producing gametes like sperm and eggs, but we're just talking about normal somatic body cells right now. And so DNA must be replicated because if you don't have instruction sets in every single cell, then the cell doesn't know what to do. So first of all, if you made it because it has to replace another cell that died, if there's no DNA in it, it can't do its job and it's pointless to have replicated it to begin with, okay? So DNA needs to be copied because every cell needs DNA inside of it. So when you split the cell into 2, each cell needs to have DNA in it to tell it what to do or what to make or how to function. And then the other thing that also needs to be in the cells are organelles. For example, if it's a plant cell, it's going to need the chloroplasts in both of the cells when it splits. It can't just have chloroplasts in one cell and then no chloroplasts in the other, right? Because both cells are going to need to be doing photosynthesis in a plant. So organelles must be doubled before they are then split evenly across 2 separate cells when that one cell becomes 2 during the cell cycle. Okay, so we're going to do like the first major portion of the cell cycle notes today, mainly focusing on cell cycle. We'll get into mitosis a little bit before we go into more detail on Wednesday. So the homework for this whole week is just to do your chapter 9 summary and Learn Smart, which is over this topic, obviously. And it is due on Friday. So I'm no longer doing Sunday due dates, I'm doing Friday due dates. And so our objective for this current unit, which is going to again span multiple chapters, is that learners will be able to describe events that occur in the cell cycle. They will be able to construct explanation using visual representations or narratives as to how DNA and chromosomes is transmitted in the next generation or to the next generation via mitosis. So first of all, we're going to start on this left side. So notes on cellular division. Prokaryotes, there are 2 kinds, 2 main groups that we talk about, and 2 main groups are actually the 2 main kingdoms of prokaryotes. And so the first kingdom of prokaryotes is eubacteria, or what we just call normal bacteria. There's also archaea or archaebacteria, and we just tend to use the terms bacteria and archaea these days in modern science. And prokaryotes mean that there is no nucleus and no membrane-bound organelles. So they don't have mitochondria, they don't have chloroplasts, they don't have a nucleus, right? They don't have endoplasmic reticulum and Golgi bodies. They can only do simple functions, but they make the best of what they have, right? So specifically for DNA, you should already know this, they have one unique circular chromosome. So they don't have those X shapes that we commonly think about when we think about chromosomes, because those are the eukaryotic organisms. Prokaryotes, like archaea and bacteria, only have one single chromosome, and it is just in one continuous loop, right? It might be tangled up, but it's just one continuous loop all the way around. So if you straighten it out, it's one, it's a circle of DNA. So it's folded up in an area in the nucleoid, right? It's not an actual nucleus, but it is kind of central to the shape of the bacteria. It's in the center-ish, so it's called a nucleoid. And it's 1,000 times the length of the cell. So if you actually stretch it out, it's really, really, really long because it's containing a lot of information. And so the ring of DNA, or the circular chromosome, must be copied into 2 separate rings before the cell divides. Otherwise, you're going to have a bacteria body with no DNA in it. So they split by a process called binary fission. It's not called mitosis, it's called binary fission. And 2 daughter cells that are made are actually identical. So it's basically like cloning. It's an asexual reproduction that is cloning. Remember, prokaryotes, like bacteria, actually get DNA variation through mutations and through ingesting DNA particles from their environment, or from ingesting particles of DNA from undead bacteria in the environment. So they're not actually really recombining their DNA like eukaryotes do, because they're doing asexual reproduction, because they themselves are splitting instead of sexual reproduction. And the reason why I'm talking about sexual reproduction, or reproduction in general right now with bacteria, is because bacteria are single celled. So they're not replacing and they're not growing by making more cells, right? Because they are that one cell. When we talk about eukaryotes, though, we're talking mitosis is similar to this process, where they are doing replacement and growth instead of sexual reproduction, because that's a whole nother story and a whole nother chapter. Okay, so if you can look at this picture on the side, it's actually already labeled. And you're going to just kind of draw a simple version of this. It doesn't have to be complex, right? Because there's no organelles or anything. But it does help that you label these things so that as you go along, you understand the different parts. So in each step, so here you can see that DNA is being doubled. And as the DNA is doubled, then you can actually move on to the next process, where the cell is going to slowly lengthen, and then the DNA is going to move to the opposite side, because you don't want the DNA actually accidentally being stuck in one cell, because then you have double of DNA, and then in another cell, you have an empty cell, which is not going to be able to live. So the DNA is going to move to the opposite ends. Then the center is going to pinch in. And then once the pinch is fully complete and the cell membrane and the cell wall are completely touching, then the cells will be able to split up, and they are identical to each other. Okay. When cells split, they're usually called sister cells. So in eukaryotes, it's a lot more complicated because there's a nucleus, like I just talked about. So in the cell cycle, it's the ordered stages to go from 1 to 2 cells. First of all, the cell has to grow larger because it needs to be containing enough cytoplasm to be split across 2 cells. It's going to be needing to contain enough organelles, and it's going to also have to double its DNA. So like I said, obviously organelles doubled, and then DNA is replicated. So this is all kind of taking up the space. So there are 2 main stages of the cell cycle. And one stage takes up most of the time, and that's interphase. And interphase has a couple sub-stages that we're going to go over. And then there's a second stage, which is mitosis, or the mitotic stage. And that is where the cell actually splits. So during interphase, the whole time, you observe the cell, but you can't actually see any differences in it. It's not doing anything that you can see with the naked eye, or even under just a simple microscope. It looks like it's just resting. But in actuality, there's a lot of stuff going on, like DNA replication and growth. But in mitosis, that's where you actually see the splitting, and you see the chromosomes actually form, because in interphase, the DNA is actually still in chromatin form, where it looks like spaghetti, and you can't really identify where each of the chromosomes are. So if you look right here, you can see that this big purple arrow is interphase, and it takes up the majority of the cell cycle. And this is actually an overemphasis of how large of a time stamp that mitosis takes inside of the cell cycle, because it actually takes about the size of just the cytokinesis area in a cell cycle. It's a very, very short time compared to the rest of the cell cycle. And sometimes cells in interphase, after they split, they just go straight into G0, which they are dormant or not growing or not replicating, and they're not preparing to split for a while. So sometimes they go into this G0 phase, and some of them don't re-enter interphase, and some of them don't and stay in G0 for a really, really long time, or forever until they die. And you have to do interphase before you do mitosis, because again, if you don't replicate your DNA, you can't actually split into 2 separate cells. And if you don't double your organelles, you can't split into 2 separate cells. So you can't just have mitosis, mitosis, mitosis. You have to have interphase in between as well. So in interphase, we're going to break it down into its 3 main phases. So again, like I said, most of the cell cycle is spent in this interphase. And again, it's not 75% and then 25% like this kind of shows. It's about 90% of this purple arrow, and then there's 10% of mitosis. So a cell's perform its usual functions throughout all of interphase. So if it's a cell that's like a muscle cell that's in charge of helping with movement, it's going to be doing that and producing a lot of ATP for energy for movement. the whole time it's in interphase. So 90% of the time, this cell is going to be in interphase and just doing its job. Nerve cells and muscle cells are actually stuck in the G0 stage. So they're not actually going to be entering the cell cycle very often. When you're younger and you're developing your nerves and you're developing your muscles, you're actually going to be able to have those cells re-enter into interphase to grow and duplicate because you're actually physically growing. But once you get to a certain age, it's earlier for nerves than it is for muscles. You actually don't grow any new nerves anymore, which is why nerve damage is a super big issue in the health industry, because once your nerves are damaged, it takes a lot of treatment to be able to have you regain feeling or regain sensory input. And sometimes it's not actually even possible because nerves don't actually regrow on their own. So inside of interphase, we have three main phases. We have the G1 phase, the S phase, and the G2 phase. And it occurs in this order where, intuitively you'd be like, G1, then G2, and then S, or S and then G1, G2. No, it's G1, and then there's a break, and then S phase happens, and then G2 phase happens. And it's just because G stands for growth, S stands for synthesis, and you're growing 2 times, and you're doing synthesis once. So in G1, the cell is going to double its organelles. It's going to grow in size, and then it's going to get ready for DNA synthesis. In the S phase, which is the synthesis phase, DNA replication or DNA synthesis actually happens here. So it happens during interphase and not in mitosis. The proteins associated with DNA are made, and then chromosomes are copied. So you're going to go from a single amount of DNA to the double amount. And usually this is done in a process that we'll actually talk about when we talk about like DNA transcription, translation. Is really closely related to that process. It's the same materials like DNA helicase opening up the DNA strands into 2 and then copying the base pairs to match these strands. And then it's like semi-conservative because half of the new strands are actually half old, but you get 2 copies of DNA at the end of S phase. And if you don't do S phase properly, then you are going to go through mitosis and you're actually going to get cells that are incapable of functioning. And then after the S phase, you're going to go into G2, and that is the last phase before you actually start physically splitting up the cell. So between DNA replication and mitosis, the cell synthesizes proteins necessary for division. So these are like cytokines that are proteins that are going to help produce spindle fibers to pull chromosomes to the opposite sides of cells during mitosis. If you don't have those, you can't do mitosis. So basically all of interphase is preparing for doing mitosis, which is splitting up. But it's most of the time where the cell is also just functioning as normal. The mitotic stage actually includes mitosis and then cytokinesis. And cytokinesis is at the very, very end. So mitosis is when the nucleus and the nuclear materials are divided, so nuclear division. So the chromosomes are being split and pulled apart into the opposite sides using a mitotic spindle, and then they will reform into 2 daughter nuclei. But then cytokinesis is actually the physical separation where the cell is going to pinch off the cytoplasm and the membranes are going to meet and they're actually going to pull apart. So mitosis is the DNA or the nucleus splitting, and then cytokinesis is actually the physical cytoplasm moving and splitting, right? Cyto is cytoplasm, kinesis is movement, okay? So in animal cells, it just pinches off right here. So it pinches smaller and smaller and smaller until it pops into 2 separate ones and the membranes are fully formed. And then in plant cells, you can't actually pinch because there's a cell wall. So a cell plate will actually form in the middle after the nuclei are separated. And then as the cell plate grows, it'll grow longer and longer and longer until it meets the top and the bottom of the original cell walls on the outside, and then it'll seal off the 2. And therefore these 2 brand new cells are actually going to be neighboring cells in the plant cell. They're not actually going to be moving around. Versus in an animal cell, for example, if you made a red blood cell, it could just float around into a blood vessel because they're not necessarily attached to each other. It just depends on the environment they're made in. So I'm going to ask that you actually go through and look up these words and write a definition for yourself. I will be checking these in your notes. You can use the textbook you can use the textbook and Control F, or you look at the vocab section, or you can just Google them and then write down a definition in your own words. So I will be checking these when I check your extra credit notes. So these also have to be done. Okay, so the last thing I'm going to ask you to do is a kind of post a project. I wouldn't really call it a project, but it's a short drawing section. So you're basically going to be drawing the phases of mitosis for me. And it's just an introduction into mitosis. So I have provided this kind of diagram for you, and I'll show it again at the end. So you're basically going to be drawing these phases with arrows like this to show me that there's a cycle forming. there's a cycle forming in mitosis, and you're going to go from interphase to prophase to metaphase to anaphase to telophase to cytokinesis, and then back into interphase. Okay, so what we're actually going to do is you're not actually going to draw these chromosomes. I want you to actually draw them for Drosophila melanogaster, which is the common fruit fly, which is a very, very common animal that we use in genetics in biology because they're easy to breed, they have short generation spans, and they're cheap. So we're going to work on 4 pairs of chromosomes, and you're going to be showing the interphase, prophase, metaphase, we'll include prometaphase since there is a diagram here for you to reference. Anaphase and telophase / cytokinesis. We'll combine those two just because in the drawing, you're going to be drawing and pinching it anyways, which is cytokinesis. So for each one, you're going to have all 4 pairs of chromosomes. They're going to be these 4 pairs. So if you notice, the X chromosome actually doesn't have the 2 parts on top. There's no upper legs on the X chromosome, and there's no upper legs on the 4th chromosome either. Drosophila melanogaster are kind of interesting because they only have 4 pairs of chromosomes. So it's also easy to tell where their genes are and what part of the genes are mutating because you only have to track 4 chromosomes. So you're going to be drawing chromosomes like this, and you can draw them into bubble-shaped chromosomes as well. So example for the X chromosome right here, you could be drawing like this instead of like the X shape, just because the X shape here is a little bit misleading. It's going to be hard for you to kind of visually represent. And for example, for the 2nd chromosome, you could be drawing them in these bubble X shapes as well. It's just up to your own preference. And I would draw the 2nd one in a separate color just because it's the one that's going to be moving away. So you could draw in a separate color if you want to, just to help you keep track instead of drawing them all in the same color. So for each phase, you must have all 4 pairs of chromosomes, right? So 1, 2, 3, 4. The chromosome should be the correct size. So the 2nd one should be bigger than the 3rd. The 4th one should be the smallest and have no upper legs or arms. And then the X should be a medium size, but they should not have upper arms either. The centromere should be in the correct location. So for the X, this blue dot needs to be at the top since there are no upper arms like in the 2nd and 3rd. Okay. The homologous chromosome should be different colors. So like I said, I want one in a color and then one in another color. So this one would be one color and then this one would be another color. But the color that this one is, it's going to be the same one that this one is and this one is and this one is. The color that's on the bottom here is going to be the same as the one over here and over here and over here. Okay. You're going to be drawing a nucleus, for example, when you're in interphase, just because, you know, you're not going through mitosis yet, so the nucleus is still there. And at the end of cytokinesis, you're going to want to draw the nucleus reforming. The cytoplasm you can leave clear. Obviously, it's inside the drawing of the cell membrane. And you're going to want to draw microtubules or spindles, like these things right here, that are helping pull the chromosomes to the correct side of the cell that they need to go on. And then make sure you just write a short description that's describing what's happening in each of the images. So I want you to describe in simple language what's happening in prophase, what's happening in prometaphase, what's happening in metaphase, anaphase, and telophase. And then one last thing that I do want you to draw that's not here in this requirement is the arrows in between the phases. I want to make sure that you know that there's an arrow going from early prophase to prophase, prophase to prometaphase to metaphase, which is not shown in this image. So I will know that you're like directly copying if you don't have, if you have this kind of alignment and you don't have an arrow connecting from prometaphase to metaphase. Honestly, it would be easier on your piece of paper to draw them in like a kind of circular pattern so that the arrows are just all going in the same direction. But it is up to you. Okay, that's for the poster. Obviously, once you finish with these notes, you can submit them for extra credits, but this poster is a separate assignment that you're going to be submitting for. And it's not really a poster, it's just a drawing. So you can take it on a sheet of paper and draw it, or if you have like an art pad that's connected to you can connect to your Chromebook, you can also draw on that. That is up to you. Just remember that you need to probably pause on this screen, and you'll probably need to pause on this screen as well as your reference. If you can find another reference for the cell cycle mitosis part, you can also use that one. So that's all for today.